Okay, welcome everybody. Let me share my screen real quick. All right, welcome to the AI in Medicine at UCLA Research Panel. I know that is quite a mouthful, but we have some great things planned for you today, including a short presentation from our speakers where they will discuss their research and tell you how you can get involved, followed by a Q&A session. And we'll finish things off with breakout rooms where hopefully you can get some FaceTime with the professors, and also you can ask any questions yourself. With that, we have created a brochure for the event, which includes the agenda, information about the professors, and the link to the feedback form, and also how to get involved. And I can send a link to that in the chat. So make sure to check that out if you can. And with that, let's get started. So quick introduction. Hello, my name is Kai. I am ACM AI's events director, and I will be one of the moderators for today, and I will pass it off to Ava.
Hi, everyone. I'm Ava, she, her pronouns, and I will also be a moderator. And I'm a member of ACM AI as well, and I'll pass it off to Professor Eskin.
Hi, I'm Eleazar Eskin. I'm the chair of computational medicine, also a faculty in computer science and human genetics. Some of you are either in my class or have taken my class before. My pronoun is he, him, and I will pass it on to Harold.
Hi there, I'm Harold, he, him. So I am relatively new faculty as of mid last year in computational medicine and human genetics, and I will pass it off to Luz.
Hi everyone, I'm Luz Odelohaus, she, her. I am computer scientist by training and faculty in the psychiatry department. And I'll pass it on to Sriram.
Hi everyone, I'm Sriram Shankararaman, he, him. I'm a faculty in computer science, human genetics, and computational medicine. And I do see some of your names seem familiar from the class that I taught last quarter. Oh, and who's going next? I have no idea.
Hi, I'll go next. My name is Samantha Andrews. I use she, her pronouns, and I am the student programs manager for the Department of Computational Medicine. And I will pass it off to Jeff.
Hi, I'm Jeff, he, him, and I'm soon to be faculty in computational medicine and involved in the research with the UCLA health system. And I think I'm the last one, so I'll pass it back.
Real quick, I forgot to mention my pronouns. My pronouns are she, hers. And with that, thank you for the introductions, everybody. So let's go ahead and get started with our presentation. So I will hand it off to Professor Esk.
So really great. It's really great to see so many people here interested in this. I'm really happy to share with you. And I'm going to give a really high level overview, kind of what is AI in medicine. Well, AI in medicine is the application of machine learning and AI technologies to health data. And at UCLA, we have a really unique, exciting opportunity to work on AI in medicine because across the street, well, it's not really across the street because we're virtual, but if we were on campus, it would be across the street. We have a health system that is really gigantic. There's 2 million individuals with electronic health records. There's 30 patients per year that have like waveform data from hospital stays. We have 40,000 DNA samples on patients, and that's going to be increasing to 150,000 soon. And every day, there's 1,500 imaging studies that are collected. And all of this data has the opportunity to do kind of AI imaging, genomics work, machine learning on medical records, and mathematical modeling. Next slide. And the way that we have organized at UCLA is really two major initiatives. One is called the AI in Medicine Initiative, which is led by Ron Halperin, which is really trying to figure out how to get these AI technologies to improve patient care. It's something that Jeff and Sriram and actually pretty much almost everyone on the call on this meeting works on. And then the other one is called Genomics plus Health, which is trying to figure out how we can use genomic information to increase health. And I'll go through some examples on the next slide. So here's actually an example of the kind of project that is going on at UCLA in this area. So many of you have had your blood pressure measured with that cuff thing that they put on your, they put on your shoulder, right? And it's a measurement, and it gives you one time point of what your blood pressure is at that moment. But in fact, if you think of your blood pressure changes. It kind of goes through a wave with every heartbeat. And what you have in the bottom is really what's called the blood pressure waveform. And there's a way to measure that using an invasive procedure where they put something into a vein. But what we've come up with is a technique that takes kind of non-invasive blood pressure cuff measurements, EKGs, and basically the waveform, oxygen waveform, and combines that using a deep neural network to be able to predict it with non-invasive measurements. So that's a kind of example of a project that kind of goes on in AI medicine. I'm going to go to the next project, next one. So another project that goes on, and this is something that actually Jeff works on, is to look at imaging data. So when there's a lot of importance in trying to understand when people have eye disease, because there's treatments that we can make, but understanding when to use a treatment for eye disease, those treatments can be invasive, even just understanding what's the, what point will someone deteriorate eye loss is a really important, important things to know for people's kind of vision health. And one of the things that happens in ophthalmology is that they have these machines like OCT imaging that collects, three-dimensional images, like very large scale amounts of data. And so a lot of what we work on is also doing kind of deep learning type techniques on these imaging data to try to predict features that are related to ophthalmology. Okay, I think next. So I want to give you, so that's just one overall, kind of just a high level of some projects. I want to kind of introduce Professor Pimentel, who can give you some more details on the kinds of stuff that he works on.
Hi there. So I would classify myself as more of a genomics person. My group is very interested in genomic technologies and so how to work with experimentalists in collaboration to actually design these experiments. So one thing we've been working on recently is how do you find the genes that are regulating the gene expression of a specific gene? It turns out to be kind of a hard problem, but we've done a lot of modeling actually from the statistical side, and then gone through and developed models that learn from existing data, and basically tried to do some optimization to find out what in practice would be the most efficient. And so this plot that you see here on the top right hand corner, or I guess middle of the screen, is basically one of the data sets. On the x-axis is actually the on the y-axis is an experiment that uses about 50 times more cells than the experiment on the x-axis, and they're actually correlating quite well. So this is actually new data that we just got a few weeks ago. But so these are the sort of techniques that we've been thinking about on how to design experiments. And if we could go to the next slide, please. The other thing we do is actually after the fact, after the experiment has been done, how do you actually infer what you measured, right? So we're trying to solve both problems from basically starting at the beginning, you design the experiment, and then after you've done the experiment, how do you actually measure or capture what you measured, right? And they turn out to be relatively hard problems because you're trying to solve an inverse problem with a bunch of things that you actually didn't observe directly. So you have to infer all these sort of latent spaces. But we've been working on this problem a lot. And what you're seeing on the bottom right-hand corner is actually the predictions that we're getting from a small data set that actually many of the top hits were validated. So these are gene interactions, gene-gene interactions that were actually validated through orthogonal experiments that are a bit more labor intensive. So the general idea is you do one of these CRISPR screens. Actually, I didn't tell you that. These are CRISPR screens. And you can literally hit thousands of genes in once to actually measure how thousands of gene-gene interactions are behaving, and then actually inferring which ones are actually doing anything. And you only do like 2 or 3 experiments, right? So rather than like actual experiments where you're doing like basically thousands in just one, right? Okay, so that's one thing that we work on. Next slide, please. And so we've been expanding a lot of these same ideas. This is actually unpublished work, what I showed you before, and this is also new stuff. So we've just started collaborating with some folks who have some really cool emerging technologies that actually aren't publicly available yet, doing some spatial transcriptomics. And the cool thing here is that you can actually measure and count direct molecules inside of a cell. So there's a few vision problems here, like finding cell boundaries and these sorts of things. But the other thing is actually tracing molecules through time points and actually being certain and mapping uncertainty in the actual molecules. And we're also trying to use these really wide CRISPR perturbations to infer entire networks, right? So we're integrating some things from machine learning, some things from causal inference, as well as some of these newer CRISPR technologies. And just to be clear, we don't do the experiments in my lab. I work with very closely with a lot of experimentalists who are actually designing and doing these experiments. So we have a lot of close collaborations in this space. And my lab does other things, but these are the ones that we're focusing on now and I think are pretty exciting. And I think that's all I have. Thank you.
All right.
Oh, yeah.
Yeah, I got it. All right. So when you think about AI in medicine and when you signed up for this event, you probably didn't think about psychiatry so much. You probably thought about like these fancy things that Eliezer mentioned or Harold, like ECG analysis or spatial analysis of images and some, AI. But you probably didn't think about psychiatry because psychiatry is about how you feel, and that may not be measured in the EHR in the same way as other diseases. And so what I'm hoping to convince you in the next 3 minutes or so is that you can also do psychiatry. You can also do precision medicine in psychiatry. And I will show you briefly one example of a particular problem that we are focused on, where we kind of do this and where we have some success in doing this. And this is studying the diagnosis of bipolar disorder. And bipolar disorder is characterized by both episodes of depression and episodes of mania. And patients with bipolar disorder, they cycle through these different ups and downs. And in clinical practice, getting to the diagnosis of bipolar disorder is actually really quite tricky, and it often takes many years from the first onset of symptoms. And so one of the reasons for this is that the patients with bipolar disorder often start their onset in the beginning with depressive symptoms. And so it's not until they have a first manic episode, so suppose here's a patient and he goes through time, in the beginning he just has depression, then he gets a manic episode at some point, and only then a diagnosis of bipolar disorder can be made. But during this time, the patient is really vulnerable and often gets suboptimal treatment that leads to an even worse outcome than if he would have gotten better treatment in the beginning. Next slide, please. And so just to illustrate how vulnerable, here are some estimates of rates of suicide attempt and suicidal ideation from the EHR. And here in the first sort of group are those patients who immediately get diagnosed with bipolar disorder. So they don't have this waiting period. And the second group are patients that are depression all along. They never switch to bipolar disorder. They're just patients with depression. And so, for example, for the stable bipolar patients, they have 15% rate of suicide attempt. For the patients with stable depression, it's 19%. Next slide. But if we look at those switchers, those patients that have to wait, their rate of suicide attempt is 36%, which is more than double than those patients that got diagnosed immediately. Just to illustrate how vulnerable the population is. Next slide, please. And so what we're doing is use all the information in the electronic health records, including their genetics, to kind of take a group of patients that start with depression and predict who will go on to develop bipolar disorder later in life. And the goal of this analysis is to actually be able to flag for a clinician, look, this patient is at increased risk for bipolar disorder, watch out for them, and hopefully reduce the time to diagnosis by months or maybe even years. And we use all the kind of information that's available in the EHR, like previous diagnosis or other demographic information, but also the information in the notes, because especially for psychiatry, you can imagine that a lot of symptoms that are relevant or behaviors are actually written down in free text rather than measures of lab values or directly measured in the EHR. And so it turns out that we're actually quite good at this. Next slide. Because here, what you can see are survival curves. So we build a predictor based on this, all these pieces of information. And then we say here, the blue line in the beginning, on the y-axis, you have how many people are still depression. And on the x-axis, you have the time as it passes. In the beginning, everybody is depression because nobody switched. And as time goes on, some people switch, and so less and less people are depression. And here, the blue line are those people that have the highest predicted probability of switching. And you can see that, and the green is this, the next tier and the next tier, and the black line is those that have the lowest probability according to our algorithm of switching. And we can see that indeed, the people that have the highest probability of switching are more likely to switch. And also they switch sooner than those from the different colors. And in fact, what turns out that based on this model, we can correctly flag 80% of patients during the first visit who will end up switching within 5 years of time. Of course, this comes a little bit at a cost because for each of the patients we flag as potential switchers later on, we actually one of them is correct and one of them is not correct. But this is really truly amazing because these patients in real life still have to wait sometimes years for an accurate diagnosis. And I think that's it.
So my group is broadly interested in understanding different types of biological data, ranging from genomics to very clinically relevant measurements. And on the one hand, we have these biological problems that motivate us. But on the other hand, we're also interested in what are the computational statistical challenges that are posed by these kinds of data. So on the one hand, we care about the questions, but we are also interested in what does it mean in terms of developing novel machine learning or statistical models and algorithms for these kinds of data. And essentially, we try to close the gap. So we want to figure out what are these challenges, be able to develop these models that are addressing those challenges, and be able to apply them to actually make sense of this data, develop insights, help clinicians make better diagnoses, and so on. So I'm really interested in kind of this entire cycle, going all the way from posing a biological or a medical question, translating it into a statistical problem. Typically, we think of these as statistical models. And then trying to figure out how do we actually apply these models to data. So that's often algorithms. That's where we as computer scientists spend a lot of our time. So once you have the algorithm, you then apply it to the data that you have. And then you ask, is it any good? Oftentimes it isn't. Your first attempt is not so good. And then you close the gap. And you do this again and again until you actually get some actual insights into the problem. And this is something that we keep doing over and over in different settings. So an example of that kind of an approach is this collaboration with our clinical colleagues from the Department of Anesthesiology in the hospital. And what they were interested in is given the information from the electronic medical record. So this was something that that Professor Eskin talked about, that we have collected, this rich data that you have across millions of patients in the UCLA health system. And now you have a patient that's walking in and getting a surgery done. Can we predict what are the risk factors that this patient is going to carry post-surgery? And one example of a risk factor is death, mortality. The good news is mortality rates in the UCLA hospital are low, but we can still do better. And the other reason why we care about this is often these patients who are at the highest risk for dying post-surgery are often the people who need maximum attention, a lot of attention from the clinicians. So if we are able to identify this a priori before the surgery from their medical record, then these would be the people who the clinicians would spend their critical resources on. So many of you have taken my machine learning class, some of you have. And what are the challenges in doing this? It turns out in the class, I talk about, you're given a matrix, a nice feature matrix of all these risk factors, and whether or not somebody is going to die. So you have a supervised learning problem, and you can make these predictions. Turns out in the wild, the story is way more complicated. And so that's where a lot of the challenges arise. You don't have this nice matrix. There are lots of missing data. The missingness has structure to it. It's not random. There is issues of whether you can even say whether somebody has died. Turns out it's actually quite challenging from the medical record. So this is where we need the help of a really interdisciplinary team here who knows both the clinical aspect of the data, but also the statistical and the computational aspect. So cutting a long story short, this was a long, intensive collaboration. We can actually develop some kind of machine learning models. In this case, it's a random forest. Turns out it does quite well in predicting mortality. It does much better than some of these scores that have been developed previously, quite a bit better. And on the right, what you'll see here is you have a risk score for some person for prior to surgery on the lower right, and then after surgery on the upper left. And what you can see is how a person's risk changes before and after surgery. For most people, they lie along the diagonal, it doesn't change much. But then you see people who move from high risk to low risk and low risk to high risk. And so now this is a surprise. The data, the model is telling us, is identifying a subpopulation. So an interesting question is, what drives these patients who switch risk? So here's a new machine learning research question. We have this black box model that is making these predictions. We'd like to go back and open up this black box. So this is a field of machine learning called interpretable machine learning. And so that's something that we've become very, very interested in trying to figure out how do we actually tease open these black boxes. Next slide. So another question that we are very interested in is understanding how our genome functions at a fundamental level. So our genomes are essentially these 3 billion long characters of A, C's, G's, and T's. And what they ultimately encode for are these traits, phenotypes. So Professor Lewis talked about depression. So that's an example of a trait that is partially controlled by our genomes, but the story is complex. So there's this function that takes your genetic variation, all the environments, and that determines your phenotype. And the question is, can we actually learn what this function is? So for those of you who do machine learning, that seems like a machine learning problem. But what's the challenge? The challenge is dimensionality. Our genome is this huge object. And so it's really hard to learn these functions. So what's changed over the last 5 years? What's happening is we are actually really getting at the stage where we can collect genome sequences from hundreds of thousands, potentially millions of individuals. So this is really allowing us to get at this function, which so far has been really hard to understand at any kind of complicated level. But there are challenges. So a challenge you can imagine is now, let's say you have genome sequences on millions of individuals, and each of your genomes is measured at millions of locations. That is an extremely large data set. So even storing it in memory is challenging. Doing complicated operations, learning machine learning models on that is even more challenging. So a question that we have been grappling with for the last many years is, how do we actually learn statistical models on these really large data sets, learn rich, expressive statistical models? And fundamentally, it's an algorithmic question. So you have to think about kind of this interplay between how complicated your model is, and then how do you actually learn this model using interesting, clever algorithms. And kind of just to tell you a little bit of the results, turns out there's a surprising finding is, although this data is very high dimensional, it has some inherent low dimensional structure. And the way you can do this, you can exploit it, is by using the power of randomization. So these are a class of what we call randomized algorithms. And here's an example where if you see the plot in the middle, so that's the blue is the method that we proposed. And you see how it scales as a function of sample sizes compared to the red and green, which were previous methods that were being used for a specific task, which is estimating how much of your trait can be explained by genetics. So it's a concept that we call heritability. So it turns out that we can actually get these models to run on these massive data sets. We can start asking interesting questions. So on the bottom figure, we try to ask whether there is some kind of interaction between whether you smoke and your genetics in terms of how much it controls the variation in different traits. So it turns out that for many traits, there is a pretty substantial impact, not just based on your genetics, not just based on your smoking status, but on this interaction. And really, this is kind of this combination of massive data sets combined with ability to run interesting models using cool algorithms. So those are the kinds of questions that we are interested in. So just to tell you, these data sets are growing even bigger and bigger. So I anticipate algorithm statistical modeling is going to be even more important in the future. So I think there's going to be lots of interesting questions to answer here. And maybe just one final comment about the growth in genomic data. There was this paper that came out many years back. It talked about how the amount of genomes that are being sequenced is growing faster, super exponentially. And they said, if you count up how much genomic data is there, it's bigger than data from, say, astronomy. And so they said, you need a new adjective. You can't just say genomic data is astronomical. You need a new adjective, which is genomical, to quantify this, the amount of data that is being generated. So the question is, can we actually make sense of all of that data that's being generated? Thank you.
Hi again. Here is how you can get involved in AI in medicine as an undergraduate. You can take our undergraduate core bioinformatics courses. Those are on the screen. They're CM121, Introduction to Bioinformatics, CM122, Algorithms in Bioinformatics, and CM124, Machine Learning Applications in Genetics. Next slide. You can join our mailing list. I send out a biweekly computational methods research newsletter, It includes interesting articles about the computational medicine faculty, details about upcoming advising and information sessions, helpful resources, and research opportunities. I will add my email address to the chat, and you can email me if you'd like to join. Next slide. You can also come to our bioinformatics minor information session next week. It's on Wednesday, April 28th from 5 to 6 p.m. Professors who teach courses in the bioinformatics minor will be talking about what you will learn in the minor and what it prepares you for after graduation. I'll also add the link to the sign up form to the chat. Next slide. You can attend one of our drop-in group advising sessions. This quarter, they're every other Thursday at noon. There's one tomorrow. At least 2 professors are available for an hour to answer any questions you may have about courses, research opportunities, graduate school, and career paths related to bioinformatics. And again, you can email me if you'd like more details. Next slide. You can apply to participate in Big Summer. Big Summer is the Bruins in Genomics Summer Undergraduate Research Program. It's an 8-week program for undergraduates interested in learning how to read and analyze genes and genomes. Our cohort is full for this year, but the application for summer 2022 will open in the fall, and I can post the Big Summer website in the chat. Next slide. Also, we are hiring. We're looking for UCLA undergraduates who want to use their coding and engineering skills to work on projects in AI and genomics. These projects can ultimately be used in your portfolio when you apply for jobs. If you're interested, you can apply to join the Computational Medicine Technology Core by emailing me your resume. And the supervisor for these positions will actually be Jeff Chang. Jeff, would you like to say a few words?
Sure, thanks. So you've all been hearing about all these great projects research projects applying AI to medicine, but there's always the additional question of how we actually bring these findings into practice or into the clinic. So with the idea behind this technology course, we're looking for some of you to join a group that'll be involved with creating software, whether that's web apps or software that integrates with the hospital system to bring these findings and deploy them into the clinic. So through this, you'd be exposed to a broad range of technologies and people, both from the front end and back end, and you'd be making a direct impact at patient care here at UCLA and possibly beyond.
Great. Thanks, Jeff. And that's it for me. So I will turn things back over to ACM.
All right, thank you, our wonderful speakers, for sharing all that information. So all the information about our speakers and everything that Samantha mentioned and Jeff is on our brochure. So for those who joined late, I sent that in the chat. And with that, we're gonna go ahead and open the chat for audience questions. Feel free to raise your hand, unmute yourself, private message me or Ava, but yeah, go ahead and ask any questions you have. And I will let Ava take over for this part.
Yeah, so Anna has a question. Go ahead.
Yeah, thank you everyone for your presentations. They were all super interesting. I was wondering, what would you say is like your biggest challenge in research, whether it's like through your collaborations with clinicians or just like the research in general?
Because I can answer from my aspect. So we work a lot with developing, statistical and statistical learning models for specific, data and genomics questions. And so we can do simulations all day long. We can, theorize about what this biological process is doing, and then the data sort of throws a wrench in it. And so I think there's a lot of sort of art in learning how to adjust and adapt these models to different data types and really learning the ins and outs of both the modeling aspect and really sort of the realities of biology, which turn out to be way more complicated than I think a lot of people give it credit for. So especially as outsiders, you know, who started in like computer science or statistics or any of these things. Does anyone else want to take a stab at that?
Yeah, I can follow up on that. I'm kind of seconding what Harold was saying. You could have a really beautiful model that you really like and that you're very proud of and that you think is going to outperform a more standard approach. But really a lot of time is spent and a lot of challenge. Like people say, noise in, noise out. Like if the data is noisy or the data is not what you would expect, you can spend a lot of time sort of trying to understand it, trying to debug it. The problem is usually not with the method, but trying to understand the truth. But that's a really good question. I had to think. I was glad that Harold said something first, so I had time to think.
So I can add it. That's a really great question. I think that, you know, one of the things that's really exciting about these projects is that there's usually very many parts, anything from getting the data, getting the collaborators, you know, getting the coming up with the kind of core kind of math of the equations that you're going to use, to, you know, running the results, making sure that you understand the results. And so I think one of the real challenges is that for things to really work, every single part of it has to work. So that makes it, you feel really good when actually it all comes together.
Yeah, I think just to add on to this, and I think this is kind of along the lines of what Harold and Lou said. Reality is complex and biology is complex, and so what that means is often we as computer scientists we don't understand most of it. So being able to come up with a problem that is both interesting and tractable requires a lot of engagement with often the domain experts, so biologists, clinicians, and that is a long process before we actually come up with a question which both is interesting and one that we are actually in a position to answer.
Thank you so much for all your responses. So next, I see Scott has a question.
Hi, first off, I want to say thank you for coming out and speaking tonight. Very great information. Great to hear from all of you. So my question was similar to that of Anna's, but pretty much if you think your response would be the same, maybe some elaboration. But I wanted to know, what was one thing you wish you would have known about incorporating AI into your field of work that you didn't know beforehand that would have helped you when you first started utilizing it?
So I can add, I came from AI actually into my field of genomics now a while ago. And I remember, when I went into it, I was really thinking about, give me the computational problem or the computer science problem, and I know how to solve it. And what I realized is that that's not really a recipe for success. I mean, the recipe for success actually turns out to be that if you really want to make an impact in medicine or biology, or even in any other area like space, space technology, anything you want to do, in order to really make an impact, you actually have to become a scientist in that area. So if you want to become as a computer scientist, make an impact in medical research, you have to actually become a medical researcher. If you want to make and that's the same for everything. So I think that's something that I wish I knew right away from the beginning.
Yeah, I can also add to this. So one of the things I value more and more and more is simplicity. So I also come from kind of a statistical background, and I love writing down very complicated statistical models and trying to figure out all of these cool math and cool kind of algorithms and to do inference in those models. And then reality hits when you actually have to use that model in your application. And time and again, I would realize that for all of the reasons we just discussed, these complicated models don't necessarily work. And that gave me a lot of appreciation for coming up with the simplest possible model and building up from there and building intuition. So having an intuition for data, which often comes by actually engaging in the field and in the problem, and building intuition is something that that I've come to value a lot.
Thank you for the responses. Does anybody have anything else to add? Okay, if not, someone messaged me and asked, I do not have many coding experience other than MATLAB, but I do want to participate in the summer program / maybe apply to a job. Would I have a chance to get in any of those, even if I have a hole in my skill set?
So I can answer that one. Actually, many of us are, I think all of us have been doing, for the last couple of months, we've been doing admissions for the summer program. So we have about, I think, more than 450 applicants. So it's very competitive. But there's a real recipe for UCLA students if you want to be competitive. If you take, you should take one of the bioinformatics core classes or machine learning. So if you take CM 121, 122, 124, or 146, and then there's a couple other options, but those are the big ones, and you do well in those classes, that's really your ticket to get in. So many times, you might not be, you haven't taken that, but next year, if you take those classes or you plan on taking them, if you can't get in, ask for a PTE to let you in. Oftentimes, we'll let you in. That's your best ticket to get into the summer programs.
All right. Okay, so if there are no further responses to that, we have a couple questions in the chat. So we'll go start off with Aisha's question, and then we'll go to Maya's question that's in the chat.
Hi, I had a question for Professor Luis. Did your model include just like information from medical records, or did it also include some kind of genetic mapping of people who were involved in the study? And also, what do we understand about the genetic markers for bipolar disorder?
Yeah, those are both good questions. And actually important to simplify things. I put all these things together, in terms of the EHR data and the genetic markers. What we currently have is one model of which I showed the performance that includes the EHR data, and in a separate set of participants, we use the genetic markers. And in the future, we're bringing them together. What do we know so far? The predictors from the EHR are much stronger than the genetic predictors, and are much stronger, are together combined, predict a larger chunk. However, the genetic markers do contribute to predicting who will switch from depression to bipolar. And actually, in the PGC, the Psychiatric Genomics Consortium, we have done a genetic study specifically setting apart cases with depression and comparing cases with depression to cases with bipolar disorder. And what we find is that genetic risk scores built from these genetic findings. So basically, you have your effect sizes from such a genetic comparison, and you can add it up and you can give everybody a score of likeliness of bipolar disorder. And those predict relatively well, but they're not yet so useful in clinical practice. In terms of what we know about genetics of bipolar disorder, the genetics of bipolar disorder is highly polygenic. And so it's not that there is a bipolar disorder gene or a mutation that gives you bipolar disorder. Rather, what it is that there are many, many variants all over the genome that sort of nudge your risk a little bit to the left, a little bit to the right. So they give you an increased risk a little bit, a decreased risk a little bit, and sort of all those variants across your whole genome together, they make an impact in terms of your risk for developing severe mental illness later in life. But it's not determining your fate. There's also environment, and so so it's a combination of these very small granular genetic risk factors and environmental impacts that happen to you that determine your overall risk profile. I don't know, that was a long answer. I hope it was.
Yeah, do you mind if I ask a follow-up, or do we have to move to the next person?
I'll let Ava decide.
We're actually going to be going to breakout rooms later, so if you have any further questions for her, then you can come to our breakout room. So let's really quickly move on to Maya's question, and that is that, oh, I lost it. How do you avoid combat the issues of bias towards/against certain communities in your models?
So I could maybe answer that one. It's a really, really great question, and that actually talks to a really exciting research area because, and actually something that UCLA has a lot of strength in. The patient population at UCLA is one of them, because of Los Angeles, is one of the most diverse in the US. And that means that the genetic study, the 40,000 individuals that they're collecting also has a very diverse set of individuals in it. And that really allows us, and there's also a really, one of the medical school is set up into themes of research. And the newest theme that they have created is one around health equity that really looks at this problem specifically. And so there's what you're touching on is a really exciting area of research that involves lots of, anything from computational people to clinicians to, patient advocates to try to get to the bottom of understanding these kind of health disparity type issues.
I can add something to that in addition to the amazing resources that are being created at UCLA, we're also specifically seeking out underrepresented populations. So for example, for our psychiatry studies, we're recruiting in Colombia and Brazil, as well as areas in Los Angeles that are specifically underrepresented in genetic studies.
I think just one minor point, well, not minor, but to add to this is that a lot of people at UCLA, in particular in the statistical genetics community, are working on trying to, there's some really big open problems about how you treat folks from different ancestries and all this sort of stuff. And I would say that UCLA is really like leading the efforts in trying to understand the genetic architecture across all populations rather than what has historically been true in the genetics community, which is almost always folks from European ancestries.
But it's a very good question and one that we all care about a lot.
All right, great. So if you do have any further questions, we will be opening up breakout rooms now. So you can ask those questions in there. And so, yeah, thank you for all your questions. Thank you for all your answers, professors. And I believe, Kai, are you opening up the breakout rooms? Okay, cool.
Yes. So real quick before breakout rooms, I just wanted to mention, if you could fill out our feedback form, that'd be great. So we can continue to hold events like this in the future. I will send that in the chat. And yes, so before I open breakout rooms, let me stop recording. So all the people who are tuning into our recording, thank you. Check out the feedback form and the brochure that will be linked in the description.
So I want to thank